Hi, good afternoon. And so I'm a faculty at Yale School of Medicine, and I'm going to talk about treatment for cancer-induced cachexia. Cachexia is characterized by anorexia and body wasting. It affects 1.3 million of cancer patients each year, and in the U.S. alone, 80% of cancer patients will suffer from cachexia, and 30% of them will die directly from cachexia. No FDA-approved drugs to treat cachexia, and there are two in phase 2 trial. One is a curcumin food supplement. The other one is a peptide inhibitor, which targets a brain area, but the target area is very deep in the brain, and then probably you have to increase very, very high dose to get in there. But what I'm going to talk about is ours will be very easy to get into the brain. And one in phase 3 trial, which is using progesterone analog, and but so far has found to induce severe blood clotting. We have developed a method to treat cancer-induced cachexia using a completely novel mechanism, and our method activates a subset of neurons in the hypothalamus called AGAP neurons. And cachexia is known to be associated with inhibition of AGAP neurons. When the neurons are inhibited, the TET3, I'm going to talk about the TET3 protein in AGAP neuron will increase, and then it will reduce appetite, increase energy expenditure, and increase stress. However, when the neurons are activated, you see the TET3 expression is decreased, and then you have increased appetite, decreased energy expenditure, and decreased stress. We have a novel small molecule called Bobcat 339, which can activate AGAP neurons, and we have a patent application with this small molecule and also its derivatives. The mechanism of action is that Bobcat induces TET3 degradation both in vitro and also in vivo in mice in AGAP neurons, degrades the protein. Importantly, when TET3 is degraded in the AGAP neurons, it activates AGAP neurons, and you want activation to increase appetite. We have validated our method in 2 mouse models. One is activity-based anorexia mouse model, and we show that a bobcat can correct 3 key features of anorexia by increasing food intake, maintain body weight, and decrease obsessive-compulsive wheel running. And also, bobcat can decrease anorexia-associated anxiety and depression, and decreases stress hormone like cortisol. Importantly, our molecule does not have liver toxicity and kidney toxicity. And we also have a ovarian tumor mouse model. We show that cancer in ovarian cancer increase TET3 expression and inhibit inactivate AGAP neurons, and our molecule can restore TET3 protein level and also reactivate AGAP neurons. We propose to use a mouse model, Lewis lung cancer carcinoma, LLC model, to further validate our small molecule. Why lung cancer? Because it accounts for almost 1/5 of all cancer deaths worldwide, and the LLC model is very well established, widely used animal model for cancer-induced cachexia. And we would like to have the 30K accelerated fund to perform efficacy study in LLC, and then to set up a stage for later PK and PD studies, and also toxicology studies of our method. The ultimate goal is to develop our method into a clinical, into a drug to treat cancer-induced cachexia. Thank you for your attention.
judges? Anyone?
Have other people tried to drug this target in the past?
No, we discovered this just for the past 2 years. We published a paper in Journal Clinical Investigation and PNAS. So we were the first to discover the molecular target and cellular target, and also first to use this in vivo in mouse model.
What measures of potency do you have for the Bobcat molecule?
Potency? Okay, so we have done this in drinking water and in intravenous injection and also peritoneal injection, 2.5 mg per kilo of mouse body weight, injection once a week, and then it has effect. And also because AGIP neurons are in the hypothalamus, it's very permeable to the blood things. So you can have a low dose and get in and then soak it up, and not like in the deep in the brain.
Perfect.
Thank you.